Thanks, Randy. So sometimes a fossil is so beautifully, perfectly, exceptionally preserved that you don't even really have to try very hard to imagine what it looked like when it was alive. This is Sinosauropteryx, which was found in China, and it's absolutely perfect. Like, you can see the outline of the body, you can see the feathers, you can see the soft tissues preserved, you can see the eyeball, you can see everything. So it's not hard to imagine what it looked like. However, as a vertebrate paleontologist, a lot of the times we don't actually find fossils that look like this. There's a reason that they're called exceptionally preserved. Most of the time, we're dealing with something a lot more like this, where we have a fragmentary bone situation, and we are trying to figure out what the animal looked like by putting a very limited amount of information back together. And so I go back to Sinosauropteryx because this is actually the last mention I will make of a dinosaur or color in the rest of this talk. It was actually this fossil and others like it that got me interested in this line of paleontology, studying molecules and trying to get an idea of how we can imagine fossils by studying them. But now I've started studying something other than color, and I'm much more interested in unexceptionally preserved fossils. So I have started looking at how we can learn more about extinct creatures by looking at fossils that are quite common, that we find often and are not particularly exceptional to look at. So I'm going to pose a question to you today, which is, if we only have a bone fragment, can we reconstruct an animal? So imagine the saddest little scrappiest bit of bone that you can imagine. Can we actually figure out what animal it came from? So when I think about the fossil record, I think about it in these three scales. The macro scale, so we're talking about a bone fragment, so it doesn't really have any discernible information just by looking at it. And then I work my way down to the molecular scale. And so that is what I will be telling you about a bit today. So if we don't have enough information looking at the bit of bone itself, looking at the macro scale, we just have to look a bit closer. So some of the molecular scale evidence that we find in the fossil record are molecules. So DNA, RNA, proteins like collagens and extracellular matrix proteins, lipids like sterols and fatty acids. I'm particularly interested in proteins as a vertebrate paleontologist because that's what you're going to find if you're finding bone. But it's important to understand how biomolecules are decaying and preserving in fossils to confidently reconstruct an animal. And that's where taphonomy comes in. So taphonomy is the study of how an organism decays and becomes a fossil. And I think about this in four rather simplified steps. Basically, an animal dies, it gets buried, you wait millions of years, and it ends up in a museum. And I think about this in three categories, and this is what my research focuses on, and it's how I break it down, is decay, preservation, and curation. So that's how I'm going to be walking you through some of the results that I have from some work that I've done so far. And molecular taphonomy really puts it all together. So that is combining the molecules and the taphonomy as the study of how an organism decays and becomes a fossil at the molecular scale. And like I said, as a vertebrate paleontologist, I'm mostly interested in proteins like collagens. And so at the molecular scale, when you're looking for collagens, you're looking for peptides, which are chains of amino acids. So I'll be talking about that. So one of the first studies that I did actually looking at decay was on a project that started in Amboseli, Kenya, on a cow. And this is actually a very special cow because it's a cow that was studied by Dr. Kay Berensmeyer when she was developing her 6 weathering stages. And this is a really iconic work that she did in the 1970s. And I like to refer to her as the queen of taphonomy because she really kind of started it all. And so her weathering stages were meant to look at modern bone and how it was breaking down so that we could see morphological changes, that macro scale, changes to the bone, and like basically look at that in the fossil record and get an idea of how it was breaking down. So she came up with these 6 stages to categorize morphological degradation in bone. And some of the things that she was interested in determining with these stages was time since death, and then also local conditions like temperature, humidity, soil chemistry. And it has very good implications for fossil studies because then if you see these structures in fossils, you can get an idea about these things. And so these are the 6 stages. As you can see, stage 1, which is in the top left, shows minimal wear, but some cracking. And then as you get down to stage 5, you get a lot more cracking, a lot more splinters. So as a molecular paleontologist, I was really interested in this study. And I wanted to examine these bones that she had used to determine these 6 weathering stages, to determine if there was a correlation between the morphological breakdown of the bone and molecular preservation. So what proteins were present and how they were breaking down in the bone. And it was such an exciting study because I don't know of any other like it. It was a project on a single animal, one cow that died in Kenya in the 1970s. And Kay started collecting the bones from this animal in 1976 through 1990. So there were 11 samples over this time period that include both surface and buried samples. And so I did proteomic analysis and found that 49 bones were present in the, what we'll call the freshest bone, or the bone immediately following the death of the animal, which is the most proteins that we found. And then all the way down to the fewest proteins that we found, which was 3 in the most weathered bone, which was from 1985 from the surface. And you can see that's not the oldest bone, but it was definitely the most weathered because it was on the surface. And then we also identified some post-translational modifications, which I'll tell you about in a sec. So to compare the weathering stages to the bones that we looked at, you can see that there are 2 actually different kinds of weathering stages that she looks at. She has carcass weathering stage and bone weathering stage. So you can see carcass weathering stage really follows the age of the bone pretty much perfectly. From 1976, you have a 1 all the way down to 1990 where you have a 5. So you're seeing the most weathering increasing with age. And then the bone weathering stage is a little bit more variable. You can see there's sort of a pattern where it increases with age, but then there's also the variability of whether the bone was buried or not, which makes quite a difference. As you can see on the right, this is a bone. These are different samples that were all collected in 1985. So this is 9 years after the animal died. And you can see there's a huge variation between the samples. There's the top compared to the middle, which was buried, and then the bottom, which is the least well-preserved sample that we looked at in this study. And you can see it's super splintered, and this is the one that only had 3 proteins left. So I compared the number of proteins to her weathering stages, and this is the bone weathering stage, and it doesn't correlate nearly as well as the carcass weathering stage. Remember, the carcass weathering stage really matched up well with the age of the bone as well. And you can see here that we go from about 50 proteins in 1976, right after the animal died, and then down to those 3 that are in the 1985 surface sample. So we are seeing that protein loss begins extremely quickly. We're talking about only, 15 years that the animal was dead and exposed for. However, one of the more interesting aspects of this is in that 1985 surface sample bone, where you only found 3 proteins, those 3 proteins were all collagen proteins. So it was 2 collagen 1s and a collagen 3. And this has very interesting implications for the fossil record, and collagens potentially being long lasting and something that you could find on longer time scales. And then I mentioned post-translational modifications. So PTMs are really just changes that are occurring to a protein. And what's cool about them is that they either occurred while the animal was alive, so you can see changes that were occurring then, and that's actually preserved in PTMs, or they are also post-mortem PTMs. So that gives you an idea of things that have happened since the animal died, so changes that have occurred on the proteins since death. And there are some proteins that are looked at to compare degradation. So carboxymethylation on lysine is one of those, and that's what we're looking at here, which is plotted to just basically show that it increases as the degradation is increasing as well. And carboxymethylation on lysine is an AGE, which is an advanced glycation end product. And the idea is that cross-linking of proteins has been hypothesized to encourage preservation on long time scales. So that might be part of the reason why. All right, and now I'm going to talk a little bit about preservation. So one of the things that I was really interested in, so when I think about decay, I think about it overlapping quite a bit with preservation as well. So I was wondering about how burial environment could influence molecular preservation. And so I decided to look at this in a group of animals that are very common, mammoths, and they all lived about the same time, but they're found in vastly different burial environments. So anything from permafrost all the way up into a bone that is fragmentary. So on the left, you see Luba, which is a baby mammoth that was found in permafrost. So it's obviously extraordinarily well preserved. However, we also find mammoths preserved just like we find any other bones preserved. So I was interested in comparing the molecular information that we found across those. So I looked at mammoths in different burial environments. Permafrost, stream deposit, which I also sometimes call a channel deposit, a hot spring deposit, and natural asphalt deposit. And they're all relatively around the same age. The permafrost has a wider range because it wasn't found in situ, so we're not exactly sure how old it was, but they're all like just close enough. So this is my data, and all you really need to know looking at this is it's a principal component analysis. And so it's showing things that are similar to one another and things that are distinct from one another. So I mentioned before that proteins are made up of amino acids. So this is based on an amino acid signature from analyzing a bunch of mammoth bones, as well as an elephant bone for control. And I also did some experiments on elephant bones where I heated them up to different temperatures to see if we would see any sort of trend in the breakdown of these amino acids that we could compare to the fossils. And so I looked at 49 amino acids and amino acid fragments, and this is how they plot. So like I said, all you really need to know is things that are similar will plot together and things that are different will plot distinctly. So here are the permafrost mammoth. It's plotting with the elephant, the modern elephant unaltered sample, as well as my lowest degree temperature experiments, which were 100 degrees Celsius. Here is the channel deposit or the stream deposit, along with the 200-degree experiments, which was like my mid-range. And then here is the hot spring deposit, plotting along with the 250-degree experiment. And up here are matrix samples that I took from the channel and hot springs deposits, which you can see are plotting distinctly from the bones themselves. So we definitely have a signature in the bone that is reflecting organic preservation, and we see quite a variation based on how it's buried and what type of burial environment. So molecular preservation does vary on relatively short time scales. We're talking tens of thousands of years based on burial environment. So curation is something I've been thinking about a lot more lately since I started working at the Cleveland Museum. And museum collections are great for doing molecular analyses. The fact is that degradation continues after a fossil is collected. And so we do still have molecular data that is probably being lost. But museums are a great source because they just have a ton of stuff. There's probably tons and tons of fossils in the museum just waiting to be analyzed. However, there are some complications when it comes to that. Mainly a lack of documentation about preparation techniques. The collection at the Cleveland Museum is over 100 years old, so there are a lot of things that have been done to the fossils that there's no way of knowing what has been done to them. And things like consolidants really make it difficult, if not impossible, to do molecular analyses. So being able to find samples that have good documentation is key. And then also destructive analyses are not as common or approved as often as I feel like they should be. And I'm not condoning destroying all of the fossils, but like I said, like a bone fragment, there's not a ton of information that you can get from it from a morphological sense. So if you can actually get a lot of data from it by destroying it, I think that is a pretty good argument to do so. So best practices for molecular analyses in museum collections is something that I'm working on now to try to get an idea of the best way to really utilize these amazing resources in what I think is a very exciting path forward for the field and learning more about ancient animals. So going back to my question that I posed at the beginning, if we only have a bone fragment, can we reconstruct an animal? And the answer is yes, but there are a lot of variables that need to be recognized. So if you find DNA or proteins preserved in a bone fragment, then there's a very good chance that those could be used to identify the animal that you have. However, you have to take into consideration what's gone on to these molecules during decay, during preservation, and during curation, probably on a case-by-case basis. So it's a work in progress, but I think that there's going to be a lot more exciting work in this realm in the future. And I will take any questions.
All right, happy to take questions from the room, and also if you're online, you can put them in the chat.
So do you think there's a better way than we're already doing to preserve fossils so that they do retain molecular sample value? If they're going to be sitting in a warehouse for several years or something before they get studied at that level?
Yeah, absolutely. So climate control is very important. That's something that we're dealing with at my museum that's very frustrating, is a lack of climate control. So keeping them at like a relative humidity that's very stable is important. And then controlling for temperature too. If everything could be kept in a freezer, that would be awesome, but it's definitely not feasible. But I think identifying specimens that are good examples or good possible candidates for molecular analysis and then treating those nicer maybe would be cool, because if you have bone fragments that you're not really going to do anything else with in the collection, definitely don't pour glue all over them, but then also maybe consider that they will be used for molecular analysis in the future.
With those ones that are preserved to that level, would you use it as what am I thinking? A time sort of thing? Or would you rather go with like I can't think of the word, like minerals to associate the time, or proteins if we have them?
I think I know what you're asking. So time is a very important factor that I didn't really get into in terms of what molecules you would expect to be preserved. So like DNA, you get about 1,000,000 years where you could expect DNA to be present, at least in a way where you could like sequence the genome. Proteins preserve longer. And so we do have these windows of time where we would expect to find certain molecules. Lipids preserve a lot longer than that. But I don't know that you would use them for time. I know like amino acids have been used for dating before, but that's sort of an older technique that I don't think people really use anymore.
So when you perform the molecular analysis, do you have to take a sample? It's a destructive process, right? Where you take a sample of the bone and crush it up and analyze it that way?
Yeah.
How much of the sample do you need to get an effective analysis?
It's not actually very much, surprisingly. So for the proteomic analyses that I do, it's about 50 milligrams of bone that I used. And so it sounds like it might be a lot, but it's really just like a little bone chunk about that big, and then you powder it up. So you don't need a ton.
Going back to your very beautiful graph, by the way, of the preservation methods, I imagine permafrost probably has the best preservation, but I'm really actually curious about places like tar pits, how that might affect preservation.
So the tar pits have notoriously awesome preservation. The problem that I had in my study was I was using surface mass spectrometry. So I wasn't extracting the organics that I was looking at. So they got completely conflated with the asphalt. So I couldn't see anything other than asphalt basically in my samples. But if you actually take the time and remove the asphalt, there's really, really nice preservation at the tar pits. Another thing, you're right, the permafrost was the best for proteins, but something that was very interesting was I also looked at lipids, and the lipid preservation was actually better in the stream deposit than it was in the permafrost deposit. So you can actually find different molecules preserving in different ways in different environments.
I know that they've done some of the studies back in like the Ediacaran. What do you think the upper limit on these protein studies are?
It's a really good question. I don't think we know still. It's something we're still figuring out. We're still collecting data. At this point, it's so like it's such a new field still that we're just kind of like collecting data and figuring out what's going on. I imagine that things preserve for much longer than we thought they would, but they do break down. So the question really becomes, how long is it useful information? So like DNA could probably preserve for a lot longer than we thought, but it's going to be so degraded that you may not be able to use it for anything.
I've actually got a question. We, as paleontologists, we rely so much on the studies from Mammoths, like, because they are so unique, like you pointed out, really amazing studies. But the equatorial savanna is just one of many potential environments. So how do you think like that protein preservation might vary time-wise if we looked at other environments?
Yeah, it would definitely vary. And there are definitely places that the preservation will be better than others. One of the best things about the Amboseli project was just the long time scale and the fact that we do still see collagens preserved, even though there's so much protein loss. So I imagine that it would be something sort of similar in a way, like you might not see the same rate of decay, but I imagine that you would still see collagens preserved in a lot of places.
Any last questions? Oh, we've got, yeah.
I have what might be a naive question, but amber, as in Jurassic Park, how good is that for preserving DNA, collagens, lipids, etc.?
So I've never gotten to study anything in amber before, which I'm bitter about. Like I really hope to get some at some point someday, but it preserves things extraordinarily well. There was a mosquito that they found blood meal still in the belly. So you could definitely get really awesome preservation in amber, but it's also extremely rare. So I would consider that to be in the exceptional preservation realm.
All right, can you hear me?
Yes.
All right, we do have one question in the chat here. Would you expect fish bones and teeth to have better taphonomic preservation than terrestrial animals?
Yes, we find a lot more stuff that was in the water than we do terrestrial animals. The terrestrial fossil record is pretty rough. So a lot of the animals that we know from the terrestrial record were buried in the water, just because preservation is so much more likely. You're so much more likely to become a fossil if you were buried in water.
An extension of that question, is it typically wind or sun that is the harshest on the preservation of surface bones?
It's an interesting question. It would probably depend on the environment. I would say temperature is probably a big one. And then humidity also probably plays a pretty big role. But that's a good question. I think that in general, getting buried very quickly helps a lot so that you're not exposed to any sort of weather aspects. Because anything, it's gonna break it down if it's not buried.